Chapter 7, Part 6. So moving on. So now we're going to talk about each of the individual pathways and kind of what's happening in each. And then what I'm going to say the byproducts or the results of going through those pathways. So all three pathways, fermentation, aerobic respiration, and anaerobic respiration, all begin with glycolysis. All three pathways begin with glycolysis. So, and what glycolysis does is turns or breaks down glucose into two copies, to pyruvic acid or pyruvates. And technically speaking, a pyruvate is not the same as pyruvic acid, but I'm not going to get technical, so I have a tendency to say pyruvate or pyruvic acid, but we're not going to get into those technicalities. I'll take either or as far as an answer, as far as what glycolysis yields. So during glycolysis, or at the beginning of glycolysis, we have a glucose molecule. So I think of it as, that grilled cheese sandwich that I talked about earlier. I chew it, break it down, it gets broken down into a molecule of glucose. From there, that glucose is broken down into 2-pyruvate. There's other stuff that happens in here. I'm not worried about that. The main point here is glucose, 6-carbon molecule of glucose is broken down into 2 3-carbon molecules of pyruvate. And again, I don't care about what happens in here. I'm not going to ask you anything about this, but it mentions it here. 3 actions alter and rearrange the 6-carbon glucose molecule, and eventually you end up with the 2-pyruvate. In this process of going from glucose to 2-pyruvate, we end up with 4 ATPs and 2 NADHs. So these are like byproducts of this reaction, okay? And of course down here it says total energy yield 2 and 2. Well, you're like, okay, well, what about this one? Well, because in order to go from here to here, we had to actually use some energy. We had to put a couple quarters in the machine in order to break down the glucose from glucose to pyruvate. So the net yield is 2 ATPs and 2 NADHs. Now, NADHs, I mentioned those earlier, and basically think of those as very important carrier molecules that we're going to talk about later, okay? The same with FADH2. And again, I'm not going to try to say those words again as I did earlier, but so at the end of glycolysis, we have 2-pyruvate, and we have 2 ATPs, and we have 2 NADHs. Now, pyruvate will either, depending on if there's oxygen present, either go on to aerobic respiration or go on to anaerobic respiration. So let me move on. But pyruvate or pyruvic acid has to be prepared for the next step in the process. So because glucose has been broken down into 2-pyruvate, pyruvate doesn't have the capability to actually enter in. Krebs Cycle cannot accept pyruvate or the pyruvic acid. So there needs to be some more catabolic activity to further break down pyruvate so it can enter that Krebs cycle. So think of a dime slot machine can't accept quarters, so we must break those quarters down into dimes, and that's the same idea as pyruvate entering into the Krebs cycle. It can't. It just doesn't fit. It's not the right component. So that means the pyruvate enters into the prep step. So remember, there's 2-pyruvate from that one glucose molecule. So the 2-pyruvate are converted to acetyl-CoA through a series of reactions, finally yielding 2 acetyl-CoA per 2-pyruvate. and 2 NADH and 2 CO2. So the two acetyl-CoA molecules are able to enter the Krebs cycle. So I kind of refer to that as the prep step. I don't know that the book calls it the prep step, but it's something that happens before the Krebs cycle, but after glycolysis in aerobic and anaerobic respiration. So what have we made? Okay, what have we made? We've made 2 acetyl-CoA, 2 NADH, and 2 CO2s. All right, so the Krebs cycle transfers the energy stored in acetyl-CoA to NADH plus and FAD by reducing them, okay? Transferring electrons to them, giving them electrons, right? So, it's just part of the process here. Along the way, the acetyl-CoA joins with a 4-carbon compound, and I'm not going to get into all of this here, but what ends up happening is in this process in the Krebs cycle, they call it spinning off, but we make some NADH and FADH2. All of the above is explained in this diagram here, and I added a little bit to it and made some things stand out. So up here was glycolysis, and here is pyruvate. All right, so like I said, you have glycolysis up here, and we have a pyruvate and a pyruvate, right? Because we have 2 pyruvate or 2 pyruvic acids that come from that single molecule of glucose. In the prep step, pyruvate, one of the pyruvates are converted to acetyl-CoA, and this happens over here. This is not just happening in one spot in any kind of organism. This happens in many different areas or many different times all at once. So this pyruvate is converted to acetyl-CoA and then enters the Krebs cycle, and then this one over here is converted to acetyl-CoA and enters the Krebs cycle somewhere else, right? So during the prep step, we get 2 pyruvate, 2 CO2, 2 NADH, and then in the Krebs cycle, each acetyl-CoA will give us 1 ATP. Remember, this is each acetyl-CoA. We had 2 here and here. So we yield 1 ATP per acetyl-CoA, 3 NADHs per acetyl-CoA, 1 FADH per acetyl-CoA, and 2 CO2 molecules. So again, NADH and FADH, they're electron carriers. This is going to be more important to understand what these guys are doing later on down the line. But in the course of a whole bunch of more reactions, you don't have to tell me all the reactions. I don't care about all the reactions. What I care about you remembering is, and I call it byproducts, but what comes off of this cycle, right? What is made in the process of acetyl-CoA being broken down into citrate, and then other intermediates, and then oxoacetate. Okay, so I'm not going to ask you about those particular things, but what comes off? A couple of CO2, some ATP, and so on. So what you end up with through the Krebs cycle is what's listed here. Per 2 acetyl-CoAs, because we started with a single molecule of glucose, and it was broken down into 2 pyruvate. Those 2 pyruvate were broken down into 2 acetyl-CoA, and then each acetyl-CoA goes into the Krebs cycle. And at the end, when we started with a single glucose, we end up with, out of the Krebs cycle, 2 acetyl-CoAs, 4 CO2s, 2 ATPs, 6 NADHs, and 2 FADHs. So later on down the line in the electron transport chain, these guys will enter into the electron transport chain, and in the process, they are like tokens. For each single NADH token, we get 3 ATP. And for each FADH token, we get 2 ATP. So, but we'll talk about that. So We went from glycolysis to the Krebs cycle, and this is what we have in the Krebs cycle. Keep in mind, you want to add that to what you got in glycolysis if you want to keep totals here. So the respiratory chain, or the electron transport chain, the ETC, okay, I a lot of times abbreviate it, the ETC. So it's a chain of special redox carriers. Now remember, in aerobic respiration and anaerobic respiration, we go from glycolysis to the Krebs cycle to the ETC, okay? We're going to talk about fermentation later. Not everybody has the ETC, meaning not every organism has the ETC. Therefore, they have other ways of making ATP. So the ETC is a chain of special redox carriers that receives electrons from reduced carriers. So these guys, as they get thrown out of the Krebs cycle, they are carriers of electrons, right? And this is important to remember, again, that those electrons are accompanied by a hydrogen. Whenever we talk about electrons, just try to remember that you have a hydrogen present with it. So these the NADH and the FADH2 are generated by glycolysis and the Krebs cycle and pass them in a sequential and orderly fashion from one redox molecule to the next. This is in the ETC. So the respiratory chain is embedded in the cytoplasmic membrane in bacteria. However, in eukaryotes, the ETC is found within the inner mitochondrial membranes. So I know, and I even put this here, this is a test question. So make sure you know where the ETC is, the respiratory chain, is in bacteria as well as in eukaryotes. The differences there as far as where it's located. So when the NADH and FADH2 make its way to the ETC, there's I think of it as, they pass along the electrons and a hydrogen comes with it, and that flow of electrons that are connected to a hydrogen down this chain it creates some energy. The chain itself is full of energy. And so when these electrons start being passed along and along and along and along this chain, and we're going to look at a picture of this here in a second, it kind of winds it up like a rubber band plane. So that allows the active transports of hydrogen ions to the outside of the membrane where the respiratory chain is located. And in the final step of aerobic respiration, oxygen accepts the electrons and hydrogen producing water. So principal components of the electron transport chain include NADH dehydrogenase, flavoproteins, coenzyme Q, and cytochromes. And they contain a tightly bound metal atom at their center that is actively involved in accepting electrons and donating them to the next carrier in the series. So the electron transport chain, reduced carriers NADH and FADH2 will transfer electrons and that hydrogen to the first electron carrier in the chain, which happens to be NADH dehydrogenase. Simultaneous with the reduction of the electron carriers, because as they transfer those electrons, remember, they're being reduced they're being reduced. So protons will move to the outside of the membrane, creating a concentration gradient. The extracellular space becomes more positively charged and more acidic than the intracellular space. So basically what happens is as these things are passed along through the electron transport chain, it winds it up. And the result is converting ADP to ATP. All right, I'm going to stop here and definitely pick up on the last bit of this in the last installment here.